Greetings everyone, I'm very glad to be with you today to talk about some of the skin microbiome changes during spaceflight. This presentation is divided into a number of short sections looking at the skin and microbiome, the interaction with the environment and stressors, changes that occur in spaceflight, some research studies, pathogen changes as well, the relationship between microbiome changes and skin responses, some summary points and some potential countermeasures. The skin is the largest organ in the human body. It forms a protective barrier against the external environment and pathogens. However, this barrier function can be disrupted, which is potentially problematic, but this function is essential for internal homeostasis or physiological balance. The skin's divided up into several layers from the outside to the inside. It forms a neuroendocrine system and also contains diverse microbial communities that form the skin microbiota. But if these are disrupted, it can potentially result in skin diseases. There is a diverse microbial ecosystem in the human body, including bacteria, virus, fungi, and protozoa. These are distributed differently and they all have their site-specific preferences and unique microbial fingerprints. There's a significant interaction between human skin-associated microorganisms and the local environment in closed systems such as spacecraft. The microbiome changes during spaceflight, which may lead to skin infections or flare-up of skin diseases. And that balance among skin microbial communities is really important. And if this is disrupted by microorganism or host factors, this results in dysbiosis, and this can then lead to pathology. The International Space Station is a closed system, and life support systems are in place to maintain conditions for humans to survive and also work productively and safely. There are a number of stresses, which are both environmental stresses and social stresses, and these can all contribute to the composition of the microbiomes and have a negative impact on astronaut health and induce skin changes. There's also isolation from expert medical care. Water supplies are very limited, so hygiene practices are different. For instance, you can't take a bath or a shower. You have to use wet wipes and wash your hair with a rinseless shampoo. There's no washing machines, so astronauts can't change their clothes as often as they do on Earth. So these factors, along with temperature changes, microgravity, radiation, and so on, do affect skin health and lead to frequent skin impairments and skin diseases. A very wide range of health issues have been reported by astronauts. But interestingly, dermatological complaints are among the most commonly reported medical events, and these can profoundly impact astronaut health, well-being, and comfort. There's a well-established dysregulation of the immune system in space. It just doesn't work as well as it does on Earth, and that's been associated with reactivation of viruses and also a persistent skin rash in one particular astronaut. And just spaceflight itself has the potential to severely disrupt the skin microbiome. So although in one sense this might seem a trivial issue, it's also something that's important to be taken into account when planning a spaceflight. A very wide range of skin issues have been reported, as well as skin damage and more chronic type skin diseases. Also interactions with spacesuits, gloves, boots, celestial dust and lunar soil can result in skin problems and infection. And a lot of this is thought to be related to the environment, including the limited skin hygiene, environmental conditions, and shifts in skin microbiota. There have been skin physiological changes noted relating to collagen production, epidermal thinning, and a loss of elasticity. However, the research outcomes have been mixed, and later research has contradicted some earlier research findings. In addition, skin thickness, barrier function and hydration differ depending on body location as well as age and gender. And a further complicating factor is that the headward fluid shift that occurs in microgravity can have an impact on skin measurements. And there was one study which looked at hair follicles, and that suggested that spaceflight inhibits cell proliferation in hair follicles. Section B of Figure 1 looks at some of the changes that occur in skin during spaceflight, including increased sensitivity and epidermal thinning. And Table 1 is a detailed listing of the skin-associated clinical symptoms and medical conditions, and you can see for that there's a very wide range of things which can occur and have been reported in space. If you have optimal skin microbiome health, then that contributes to the skin barrier working well along with skin immune function and healing. However, if there's dysbiosis, then you're likely to have changes in pH, changes to dermal immune function, and the possibility of chronic skin conditions. And in spaceflight, there are multifactorial challenges to skin health and to the skin microbiome. And all of those, when you combine them together with the potential increased virulence of viruses and bacteria, has overall implications for skin health and astronaut well-being. There is existing terrestrial evidence to suggest that many health problems suffered by astronauts can be caused or exacerbated by microbiome dysfunction. There was a research study which went over 33 months looking at the microbiome on the ISS and astronauts in the ISS. And this observed alterations in the skin microbiome that might contribute to the high frequency of skin rashes and hypersensitivity experienced by astronauts in space. There's good evidence to show that there's a rapid exchange of pathogens between human beings and the environments that they're in. And the ISS is a closed environment. It has its own microbial community, receives cargo shipments which have a low microbial load, and of course astronauts who have a comparatively high microbial load. And the Voorhees study looked at the microbiota of 6 ISS surfaces and how they compared to the microbiomes of the crew. And there was quite a bit of overlap, but the ISS microbiome did change significantly over time. The research showed that although there were significant changes over time, some bacteria were there all the time, and that the correlation between the ISS samples and the astronaut samples tended to converge over time. Something else that they noted was that the response of the skin microbiota was very individualized, with going up in some and down on others in terms of diversity. The study also showed that a very wide range of bacteria and fungi play a role on the International Space Station, and it is of concern that some of these do show some antibiotic resistance. 2 other studies of interest have shown that there is an association between being in the natural environment and the abundance of skin proteobacteria, which do seem to have a protective effect as far as the skin is concerned, and also that astronauts can inadvertently bring in environmental fungi. So this is certainly something to keep an eye out for, and is also something which will need to be vigorously monitored for in off-world environments. The Voorhees research study showed that there were some microbes which had decreased abundance in space, some which had increased abundance, and that there were changes from pre-flight to in-flight and also post-flight. The changes in the skin microbiome are likely to be multifactorial. However, it does appear that the reduction in proteobacteria can be linked to the high frequency of skin hypersensitivity reactions, rashes, skin infections, inflammation, and allergy sensitization. And it's also thought that the change in skin structure might facilitate the establishment of opportunistic skin infections, for instance, by staphylococcal and streptococcal species. An increase in the Malassea fungal species does seem to be linked with poorer hygiene, and it's also linked to dermatitis. So this could be an explanation for why astronauts might experience more dermatitis type symptoms. Also a suggestion that changes in bacterial skin communities could be linked to the use of certain synthetic garments on the ISS. The key points to come out of the studies do seem to be that variability in the skin microbiome does tend to decrease in space, and that living conditions and skin hygiene are different in space, and that this can affect how the skin reacts and the likelihood of developing skin hypersensitivity reactions and infections, and that there is an ongoing exchange of microorganisms between astronauts and the confined environment. Some further take-home points are that it's important to understand the skin microbiota changes that occur in spaceflight to maintain the balance of skin microbiota and to do more research about what's likely to happen to skin with very long duration space missions. In terms of countermeasures, it's important to note that optimal skin health occurs within a range of pH values, and having the right sort of pH helps to stabilize the human microbiome ecosystem, and in turn the microbiome is greatly affected by pH and changes in pH, and common pathogenic bacteria have their own preferences for optimal growth at a particular pH level. An acidic pH is important for microbiome health, and in higher or more alkaline pH, the microflora will change its composition and allow certain bacterial and viral species to become more virulent. It's thought that there would be multiple benefits of maintaining a lower skin pH in microgravity, including maintaining skin barrier, reducing dryness, reducing infection risk, slowing disease development, and increasing immune responses and wound healing capabilities. This could be as simple as using slightly acidic wipes for washing, maybe 3 times a week. A key takeaway point is that improving and maintaining skin microbiome quality will reduce the risk of skin dysbiosis and infections during long-duration spaceflight, and management principles should include correcting pH, balanced level of fatty acids, and avoiding substances that damage or disrupt the microbiome. Elderly skin tends to suffer from many of the same complaints as astronauts during spaceflight, and treatment here also suggests appropriate lowering of pH in skincare products. However, there are challenges for medications and treatments during spaceflight, including manufacturing, medication stability, and storage. Some other suggestions for optimizing healthcare in space include best practice body cleaning methods, using moisturizers, and addressing improper spacesuit, glove, and boot fit induced skin breakdown. The ventilation system needs to be able to deal with all the little skin particles which are floating around in the air. And on a more serious note, immunodysregulation does mean that astronauts are more susceptible to infections, so you have to look at the preventive side of that, including vaccination and screening astronauts for Staph aureus, both antibiotic susceptible and antibiotic resistant varieties, and decolonizing them as appropriate. And there's also recent research which suggests that colonizing people with Staph epidermidis might selectively inhibit Staph aureus colonization and biofilm formation. So that could be an interesting countermeasure in itself. Thank you very much for your attention.